What is protein synthesis? The information content of DNA is in the form of specific sequences of nucleotides along the DNA strands. The DNA inherited by an organism leads to specific traits by detecting the synthesis of proteins, and the process by which DNA directs protein synthesis. Gene expression included two stages, called transcription and translation. But what it consists? The transcription is the synthesis of the RNA under the direction of DNA, produces messenger RNA. The stages of the transcription are initiation, promotes signal in the initiation of RNA synthesis and help eukaryotic RNA polymerase recognize promoter sequences. Elongation, RNA polymerase synthesizes a single strand of RNA against the DNA, template strand adding nucleotides to the tree end of the RNA chain. As RNA polymerase moves along the DNA, it continues to untwist the double helix, exposing about 10 to 20 DNA bases at the time for pinning with RNA nucleotides. Termination, specific sequences in the DNA signal termination of transcription.
Post-termination RNA processing. Most eukaryotic mRNAs aren't ready to be translated into protein directly after being transcribed from DNA. mRNA requires processing. Transcription of RNA processing occurs in the nucleus. After this, the messenger RNA moves to the cytoplasm for translation. The cells adds a protective cap to one end and a tail of AS to the other end. These both function to protect the RNA for enzymes that will degrade. Most of the genome consists of non-coding regions called introns. This, an RNA copy of a gene, is converted into the messenger RNA by doing two things: add protective bases to the ends and cut out introns. Alteration of mRNA ends. Each end of the pre-mRNA molecule is modified in a particular way. The 5' end receives a modified nucleotide cap, and the 3rd end gets a poly A tail. RNA processing splicing. The original transcript from DNA is called pre-mRNA. It contains transcript of both introns and exons. The introns are removed by a process called splicing to produce messenger.
Translation is the RNA direct synthesis of a polypeptide. Translation involves mRNA, ribosomes, ribosomal RNA, transfer RNA, genetic coding codons. Genetic code. Genetic code information is encoded as a sequence of base triplets or codons. Codons 3-base code for the production of a specific amino acid sequence of 3 of the 4 different nucleotides. Since there are 4 bases and 3 positions, there are 64 possible codons, but only 20 amino acids. 3 of the 64 codons are used to stop signals. One code is used as a start signal. Transfer RNA consists of a single RNA strand that is only about 80 nucleotides long. Each carries a specific amino acid on one and has an anticodon on the other one.
Building a polypeptide. Initiation brings together mRNA, tRNA, bring the first amino acid of the polypeptide and 2 subunits of a ribosome. Elongation. Amino acids are added one by one to the preceding amino acid. Termination. The final stage when the ribosome reaches a stop codon in the mRNA. Translation. When the ribosome reaches a stop codon, there is no corresponding transfer RNA. Instead, a small protein called release factor attached to the stop codon. The release factor causes the whole complex to fall apart. Messenger RNA. The 2 ribosomes submits the new polypeptide. The messenger RNA can be translated many times to produce many protein copies. Post-translation. The new polypeptide is now floating loose in the cytoplasm if translated by a free ribosome. Sometimes other molecules are also attached to the polypeptide: sugars, lipids, phosphates, etc.